Hi, this is Aryan, and this lesson will be on cell structure. But before we get to actual cell structure, we need to start with the basics on cells in general. So, cells are the smallest unit of a living thing, and the vast majority of life is made of single cells, like bacteria. But all life you've seen day to day is probably made of many, many cells, usually upwards of a few trillion. So the way we can view these really small cells with a microscope is with a microscope. And in microscopy, there are two really important values. Magnification is how much you can enlarge an object, while resolution is how clear the details of an object are. So it turns out that there are actually a bunch of different types of microscopes. First of all, there are light microscopes, which are used in most teaching settings and can magnify up to 1,000 times. However, scientists often use electron microscopes, which visualize much smaller objects using electrons rather than light. And this results in much higher magnification, but because the light is not used, the resulting images are in black and white. If you've ever seen a colored image from an electron microscope, that color has been added in later and is therefore called false color. It's generally used to differentiate structures that are hard to tell apart in black and white. Okay, now let's move on to actual cell structure. First of all, there are two types of cells, prokaryotes and eukaryotes. The main difference between the two is that prokaryotic cells do not have a nucleus, while eukaryotic cells do. Eukaryotic cells are generally considered to be more complex and often make up multicellular organisms like plants, fungi, and animals. However, there are single-celled eukaryotic organisms. For example, the germ that causes malaria is a single-celled eukaryote called Plasmodium. On the other hand, all prokaryotes are unicellular, and this basically covers all bacteria. Cells are, of course, really, really small, but it turns out that prokaryotic cells are actually significantly smaller than eukaryotic cells on average. And being small is often advantageous for prokaryotes because it means they require less nutrients to survive and don't have to worry that much about transport within the cell. On the other hand, as you'll see in a minute, eukaryotic cells need extensive networks in their cell just to make sure that everything is going where it needs to go at the right time. Okay, so now let's start with eukaryotic cell structures. First of all, these little cell components are called organelles, and each one of them serves a really specialized purpose. The first one I'll be going over is the cytoplasm, which is really just everything inside the cell. It's made up of the organelles, the space in between them, which is filled with a gel-like substance called cytosol. Now we're going to move on to the nucleus, which is the main organelle that differentiates eukaryotes and prokaryotes, and its main purpose is to house the cell's DNA, which is the macromolecule that I showed you in the last lesson. Now DNA is the genetic code, which means that it really holds all the information that the cell needs to grow, survive, and coordinate with nearby cells, pretty much everything that's associated with life. And I'll get to how and why we need DNA to do all this later in this course, but for now it's just really important to remember that the nucleus is filled with really, really compacted DNA, and that this DNA is called chromatin. Now this chromatin is packaged incredibly tightly using special proteins, and then wound around itself until it forms chromosomes, which you can see in this picture. Now this is a eukaryotic chromosome. Prokaryotic chromosomes are not packaged densely at all. They're basically just really big circles of DNA. Also, remember that prokaryotes have no nucleus. They don't have to worry about keeping all their DNA in one very small part of the cell. The nucleus is also filled with a gel-like substance, which is called the nucleoplasm, and is surrounded by two membranes, both of which are similar to the cell's membrane, which I'll go over later in this presentation. These two membranes are called the nuclear envelope, and their main function is to prevent RNA, proteins, and small molecules from crossing over from the outside to the inside, and from the inside to the outside in the case of DNA. The nucleus also contains this other structure called the nucleolus, denoted by the filled circle in the nucleus in my drawing. And the nucleolus is where ribosomes, the organelles that produces proteins that I drew as these small red dots, are made. Now, ribosomes are made of both RNA and proteins, which come together to form some really complicated machinery, and then these ribosomes are then shipped out of the nucleolus into the nucleus and then eventually into the cytoplasm. Okay, so now let's go over ribosomes. Compared to other organelles, they're really small, but of course really important. Basically, RNA is copied from DNA in the nucleus and then brought out to the ribosomes in the cytoplasm. And then the ribosomes read the RNA and use it to make proteins out of individual amino acids, which I showed you in the last presentation. Proteins are then involved in pretty much every biological process, so there are always a ton of ribosomes all over the cell, as you can see. Okay, so the next organelle I'll be going over are vesicles, also known as vacuoles in some cases, which I denoted by a gray circle near the top of the cell. These are basically containers that the cell can use for a variety of reasons, depending on the exact role or lifestyle of the animal cell. These vacuoles can be used to store anything from nutrients to a protein that the cell is specialized to produce. And there are a lot of different ways vacuoles can be converted to other organelles. For example, the peroxisome, which I drew as small pink circles. These peroxisomes are really just smaller modified vesicles that contain special enzymes that are used to break down and recycle molecules like fatty acids and amino acids. However, when these substances are degraded, the byproducts are H2O2, better known as hydrogen peroxide. Now, hydrogen peroxide is really dangerous to have in your cell just floating freely around. So after the peroxisomes are forced to make it when they recycle other stuff, they then safely break it down into oxygen gas and water using enzymes. Vesicles can also be used to make an organelle called the lysosome, which are these dark green circles in my drawing, and lysosomes are basically like the garbage disposal of the cell. They use a different set of enzymes to break down pretty much anything that the cell no longer has a use for, and this can include proteins, lipids, nucleic acids, polysaccharides, or even entire organelles. Lysosomes have pretty acidic contents, which helps the enzymes in them with the breaking up of the cell's waste. Alright, now let's move on to the centrosome, which I drew as light green tubes here in the center. It consists of these two tubes called centrioles and helps the cell set up for division by moving all the different parts of the cell around so they can be split evenly. They also replicate along with the cell, so each daughter cell will also have two of them. And I'll explain their role in more depth in my mitosis and meiosis presentation later in this class. Alright, so now we're going to move on to the endomembrane system, which is a group of organelles that basically are just stacked up layers of membranes. And the first one I'll go over is the endoplasmic reticulum, often abbreviated as the ER, which is the dark blue layered organelle that's connected to the nucleus in my drawing. Basically, it helps modify proteins and make some lipids. It can be further divided into two sections called the rough ER and the smooth ER. In the rough ER, ribosomes are embedded into the folds of the membrane, and these ribosomes synthesize proteins into the rough ER, where they're modified to function better in their target location, which may be in the cell, outside of the cell, or even in the cell membrane. And once these proteins are modified, they are shipped to wherever they need to be when part of the RER's membrane buds off, forming a transport vesicle. Alright, so the counterpart to the rough ER is the smooth ER, which has a ton of roles. What differentiates it from the rough ER is that it doesn't have any ribosomes embedded in its membrane, so it doesn't work as much with the modification of proteins. Like I said earlier, some types of lipids are made there, but it can also be used to detoxify substances or synthesize certain carbohydrates, and in muscle cells, it's modified to form a different organelle called the sarcoplasmic reticulum, which contains a ton of calcium atoms, which it can release, and when it does this, it'll trigger a coordinated muscle contraction among many different cells. Alright, next are the mitochondria, which I drew in light blue here. They also play a pretty big role as the cell uses them as part of cellular respiration, which is a process I'll go over in much greater detail later on in this class. But basically, in it, glucose is turned into an energy source, and all eukaryotic cells use mitochondria for a part of it. Okay, so the next part of the endomembrane system is the Golgi apparatus, which is where all the proteins from the endoplasmic reticulum and transport vesicles go to. The transport vesicles fuse with the Golgi membrane, releasing their contents, which are usually proteins and lipids, into the Golgi apparatus. And in the Golgi apparatus, these proteins and lipids are further modified so that they can be fully functional. And most importantly, they are tagged with a small molecule and sorted by target location. These sorted and tagged proteins and lipids are then packaged into secretory vesicles that butt off of the Golgi apparatus's membrane and go wherever the now sorted lipids and proteins are targeted to. Okay, so the next set of organelles are called the cytoskeleton. And while many cells have this normal kind of globular shape, many cells need to structure themselves to fit in perfectly into their niche. And this is done using the cytoskeleton, which you can think of as a network of different types of fibers. And although it serves more than just for structural purposes, it's also how the cell moves things around inside it or changes its shape. However, they aren't all the same type of fiber. Specifically, there are microtubules, microfilaments, and intermediate filaments. Microfilaments are usually found around the periphery of the cell, while microtubules are found mostly in the center, and intermediate filaments are found pretty much everywhere. So let's start with the thinnest type, the microfilaments. They're basically a very long string of actin proteins and are really easy to lengthen and shorten because of how simple their structure is. Here's a picture of one on the screen now. And they play a pretty big role in cell shape, especially for cells that are going to rapidly need to change their shape. For example, if you've ever seen an amoeba, you'll notice that they change shape really quickly in order to move around and extend parts of their cell to grab food. This is all done by microfilaments growing and shrinking extremely quickly. All right, next are intermediate filaments, which are shown here. These aren't as skinny as microfilaments and are a lot harder to unwind. However, this means that they're really good at withstanding tension. Because of this, they are usually used to maintain structure and anchor down organelles instead of for movement like microfilaments. Also, the intermediate filament pictured here isn't the only type. There are actually a bunch of different ones, and they all serve similar roles, so they're grouped together under this one name. Okay, next up are microtubules. These are by far the largest cytoskeleton component, and this is because they're hollow. I don't know if you can tell from this image, but microtubules are basically just a hollow tube instead of a simple fiber like the other cytoskeleton parts. They're made up of two types of proteins, which are alpha and beta tubulin, represented as pink and blue balls in this image. They're really interesting because, like microfilaments, they can grow and shrink, but they also have a degree of stability because of how wide they are, and therefore it takes much longer for them to change size, which then makes them much more like intermediate filaments. They help with moving chromosomes during cell division. They also provide a kind of scaffold to move things in the cell generally, like transport and secretory vesicles, for example. And lastly, they make up structures that help some cells move extremely rapidly, namely cilia and flagella. This is a really old depiction of organisms that use these organelles. The one on the left has cilia, which are the very small, short protrusions from the top of the cell, and the one on the right has that one large flagella. And these organelles can be used for a variety of purposes, often just to move the cell, but also if the cell is held stationary, for example in the human body, they can help to move fluid around. And this is kind of a tangent, but cilia are actually used in the very, very early embryo to create left-right asymmetry. So, for example, you have different organs on your left and right side. A great example is your heart is only on the left side of your body. And the development of these differences between the left and right side start really early when cilia are organized to sweep a fluid which likely contains vesicles with specialized proteins inside them to only one side of the early embryo. And scientists found this when they disabled certain proteins that cilia need to function. This resulted in randomized asymmetry. So basically half the time mice would be mirrored as compared to normal. And it's not really important that you know this, but I just think it's really cool as it's a really unique example of how cilia are used even in developmental biology. Alright, now let's test your understanding of animal organelles with this quick practice problem. if an animal cell has a non-functional endoplasmic reticulum, which two organelles will be affected the most in the short term? And I asked for two organelles, so this is a multiple answer, multiple choice problem. I'm going to give you a few seconds to work through this, and then you can pause the video in that time, and I will look through every single answer choice after that. Okay, hopefully you've taken the time to read through all the answer choices, and now let's take a look at them. So before we consider them, it's important to remember the function of the endoplasmic reticulum, which is to produce and slightly modify proteins and lipids. The pretty clear answer here is the Golgi apparatus, as it's just the next step in the endomembrane system. But the second choice was a little harder. The answer was actually the cytoskeleton. As we just went over, the cytoskeleton is a pretty big and dynamic organelle, and every part of it is made up of different types of proteins, specifically actin and tubulin in the case of microfilaments and microtubules. So if there was no endoplasmic reticulum, then these proteins and the many different ones used in intermediate filaments couldn't be modified in order to become functional in the cytoskeleton. Let's go over a few of the answer choices. The centrosome, while being made of protein, only really divides and therefore needs a new protein when the cell as a whole divides, so that likely wouldn't be an issue in the short term. Then there's the nucleolus, which serves the function of creating ribosomes and their parts, both in terms of ribosomal RNA and the proteins used in ribosomes. And the nucleolus doesn't really use proteins from the rough ER. Actually, I take that back. It does, but those proteins have a really slow turnover, so it wouldn't really be hurt in the short term. And then ribosomes really only use components made in the nucleolus, so they also wouldn't have an issue. And of course, in the long term, the lack of an endoplasmic reticulum would ultimately shut down the cell and kill it, so all these organelles would be non-functional, but this question really only asked for the short term. All right, let's move on. So I think that covers pretty much everything for animal cell organelles, so now we'll move on to what makes plant cells different. All right, so this is a plant cell. I'm sure there are a few things that really stick out to you initially. One being that there's a really thick cell wall outside of the plasma membrane that's also found in animal cells. There's also these green organelles that look similar to mitochondria, along with these small green dots in the cytoplasm, and a really, really big vacuole that takes up most of the space in the cell. You'll also notice that some organelles from the animal cell aren't present in the plant cells, specifically the centrosome, lysosomes, and microtubule structures like cilia and flagella, even though I didn't actually draw them in. By the way, plants still have microtubules, just not the structures that result in motility, because plant cells don't really need to move. Okay, so let's take a look at these structures that are unique to the plant cell. First of all, the central vacuole is this massive vacuole in the middle here, and that's used by the plant cell for a bunch of different purposes. Primarily, it supports the cell by having really high pressure, keeping the cell wall stretched tightly enough so the cell can maintain rigidity. However, the vacuole is also used to store water and nutrients, so when the plant runs into tough times, it'll resort to using this vacuole to stay alive, reducing the pressure on the cell walls, which we see as wilting. Alright, next are these small green dots, which are scattered around the cytosol. These are called plastids and are basically just a source of pigment. Finally, we have mitochondria-looking green organelles, and these are the site of photosynthesis, which is a process I'll go over later, in which sunlight and CO2 are converted to oxygen and glucose, and these are called chloroplasts. They actually have their own DNA, just like mitochondria, which is a pretty big similarity, and they're also their own ribosomes, which makes them, and these ribosomes are really different from eukaryotic ribosomes too. And because of these observations, scientists have developed something called the theory of endosymbiosis, which states that chloroplasts and mitochondria are the result of bacteria being engulfed by a eukaryotic cell. But by chance, instead of being digested by a lysosome, these bacteria began to work with the eukaryote, which turned out to be a mutually beneficial relationship, also known as symbiosis. This makes a lot of sense, as mitochondria and chloroplasts are similar size to bacteria. They have their own DNA that's in a circular chromosome, which is separate from the eukaryotic DNA, and they even contain ribosomes that are structurally far more similar to prokaryotic than eukaryotic ribosomes. Alright, next we'll be taking a look at the cell membrane, an organelle that I kind of ignored when going over prokaryotic, animal, and plant cell structures because there's just so much to say about it to the point where I'm just going to deal with it in a completely discrete part of the lesson. So first of all, biological membranes at their simplest are made of a phospholipid bilayer. Now what does this even mean, right? Let's start with the phospholipid part. If we break down the word phospholipid, we get the two halves phospho and lipid. So obviously this is a lipid, which I went over in the macromolecules part of the biochem presentation. And here's an example of a phospholipid. As you can see, there are these two tails on the left here made of carbons and hydrogens, which makes them really, really non-polar, which means they don't interact with water. While on the right here, we have this thing called a phosphate group, and it consists of a phosphate and 4 oxygens. You'll notice that one of the oxygens has a negative charge, which means that this side of the molecule is polar, so it will interact with water. So if one side is polar and the other side is nonpolar, we call this an amphipathic molecule. And now if we take a look at the rest of the phrase, bilayer implies that there are actually 2 layers. And this makes sense if we consider the amphipathic nature of phospholipids. Basically, because water is polar, the phosphate part, also known as the head, will face outward, while the hydrophobic nonpolar tail-like lipid group will kind of group together and form the inside part of this bilayer. And as you can see on this slide, and for now only focus only focus on the gray phospholipid part. We'll get to everything else here in a minute. Anyway, this is what's meant by the phrase phospholipid bilayer. These are the 2 opposite layers of phospholipids with the hydrophobic tails in the center and the hydrophobic heads on the outside. Or rather, the hydrophilic heads on the outside. And now you can see that there's a lot of other stuff involved in the membrane, and that's because the phospholipids aren't really connected to each other, so all these other components of the membrane can move around as if the bilayer were a fluid. Therefore, we call this the fluid mosaic model of the membrane. And before moving on, I'd really like to go over all the places in which we find the phospholipid bilayer in eukaryotic cells. First of all, obviously, the overall cell membrane, also known as the plasma membrane, is a phospholipid bilayer. Then there's the endomembrane system, which consists of the smooth endoplasmic reticulum, rough endoplasmic reticulum, and Golgi apparatus. And then we have different organelles, which are used to hold things. So this would include vesicles, vacuoles, peroxisomes, lysosomes, and of course we have to include mitochondria and chloroplasts, as according to the theory of endosymbiosis, they were at some point free-swimming prokaryotes with their own cell membrane. And finally, the nucleus has not 1, but 2 phospholipid bilayer membranes that come together to form the nuclear envelope. So that really goes to show how essential the membranes are to life as a whole. Okay, so let's go over the parts of this drawing that form the mosaic part of the fluid mosaic model. First of all, integral proteins are proteins that go all the way through the phospholipid bilayer. However, this means that they also need to be amphipathic in order to fit in. So usually they have a nonpolar central region with polar regions on the periphery. They can also cross into the membrane many times, which I didn't draw in my model of the membrane here, but if you can imagine using a sewing needle to weave a thread in and out of fabric, that's another shape an integral protein could take. Integral proteins usually function as receptors for signals from outside the cell, or they can even serve as channels that allow a certain type of atom or molecule into and out of the cell. Then there are proteins that are still embedded into the membrane but don't go all the way through. These are called peripheral proteins, and I drew in 3 of them into my membrane here in the middle and right side. They serve a more limited series of roles, such as proteins that identify the cell or connect to parts of the cytoskeleton. Carbohydrates are also a pretty common component of the phospholipid bilayer. I drew them in red. But unlike proteins, they're always attached to something. They can be found bound to phospholipids, forming what are called glycolipids, or to peripheral proteins, forming glycoproteins. Okay, the next part of the mosaic is cholesterol. Now, technically, cholesterol is also a lipid, but at the end of the hydrocarbon tail, there are these 4 carbon rings, just like how phospholipids have that phosphate group at the head of their lipids. Except the 4 rings are still hydrophobic, unlike the phosphate head. This results in cholesterol being incorporated into the hydrophobic center of the bilayer. Now, you may be wondering, what does cholesterol even do in the middle of the membrane? It doesn't really have access to the inside of the cell or the outer environment. Well, it does have the really interesting property of reducing a membrane fluidity. And membrane fluidity is basically how easy it is to disrupt the membrane. For this to make sense, you have to kind of understand that the membrane isn't explicitly being held together by covalent bonds. Instead, it relies on the hydrophobic phospholipid tails grouped together and hydrophobic heads that interact with the water on the outside of the cell. And while this is pretty reliable as a bond at temperatures near our body temperature, which is generally 37 degrees Celsius or 98 degrees Fahrenheit, as you get way hotter, the membrane will eventually actually disassemble. And as you get way colder, it'll become really rigid to the point where it can lose all its fluid properties, which may cause a ton of problems for the cell because it's pretty much impossible to move around or do anything with vesicles at that point. So what cholesterol does is it really just increases the range of temperatures that the cell membrane can function normally at. So like I said, we have integral proteins, we have these peripheral proteins, there's sugars, which I drew in red here, and cholesterol, which are the 4-loop lipids in orange that take residence in the center of the bilayer. Okay, so let's finish up with one last big multiple answer, multiple choice question. So many of these organelles will be the correct answer. And the question is, which of the following organelles are not membrane bound? And once more, I'm going to give you a few seconds. You can pause the video, go through this, answer for yourself, and then I will go through each of these answer choices one by one. Okay, now let's go through this list one by one. Microtubules are basically just made of tubulin, so they aren't membrane bound, so that's a correct answer. The nucleus is definitely membrane bound. In fact, it has 2 membranes forming the nuclear envelope. The nucleolus is just a region in the nucleus dedicated to ribosome production, but it isn't actually membrane bound by a specific membrane. So that's also a correct answer. The Golgi is part of the endomembrane system and is therefore membrane bound. Lysosomes are basically just a membrane-bound sac with enzymes in it. Mitochondria definitely have membranes. The rough ER is also part of the endomembrane system, so it's definitely membrane-bound. And now microfilaments, like microtubules, are really just strings of protein. So in this case, they would also not be membrane-bound and therefore are a correct answer. The central vacuole is just a huge membrane-bound space, and chloroplasts are obviously membrane-bound, as they likely came from an independent prokaryote according to the theory of endosymbiosis. These are the references for some of the images I used in my slides, and that about wraps it up for this lesson. Next time we'll be going over mitosis and meiosis.